And I'm going to introduce our last speaker for this session. That is Arpana Agrawal, who is a professor of psychiatry and the co-chair of the substance use disorders working group of the PGC, and has really been driving a lot of efforts in this area. She's based at Washington University, and we're thrilled to have her conclude this session.
Thank you, Jordan. And thank you so much to Jerome and Topher for organizing this symposium and for inviting us to present. So my task today is to give you a quick overview of what the Substance Use Disorders Group has been up to. I am joined by Howard Edenberg and Joel Galernter. We are together the co-chairs of the PGC Substance Use Disorder Group. And let's see here. This slide you've seen before if you were in Pat's talk. Just gratitude to everybody within the PGC, all of our data contributors. And a special thank you to the analysts who work very closely on the Substance Use Disorders Group. This is a list of all the individuals who contribute. Many of them self-fund themselves via NIH grants. And we are supported not just by the NIMH, but also by the National Institute of Drug Abuse that actually pays for our work. Let me start by asking why we study substance use disorders. It might seem obvious, and perhaps it is, but we are studying one of the more prevalent psychiatric disorders in the community, also amongst some of the leading causes of preventable death in developed countries. Substance use disorders occur quite comorbidly with many conditions, and they tend to exacerbate the course of those conditions. They're moderately heritable, so that's interesting for us. The other thing that you'll notice as we walk through those slides is that substance use disorders create for a fascinating case study in that they have a very distinct set of stages that undergird them. So there's an impulsivity component at the earlier stages and a compulsivity component that kind of follows. And what we're beginning to wonder is if these stages of use and misuse might actually be etiologically distinct. And I'll show you something along those lines. And of course, much like the many psychiatric disorders and phenotypes we've been talking about, substance use disorders are actually very hard to treat. So the work that our group has been doing has really been focused on three domains. This is our first cycle of funding through the PGC. And our first aim is to identify actionable variants that are associated with substance use disorders. We're very specifically interested in problematic aspects of substance use. So not so much the typical consumption variables, but more so when people experience loss of control, when there's psychopathology involved. And we want to do this at the level of individual drugs as well as for polydrug signals. Because there are these distinct stages, we're also interested in outlining the differences between genetic signals that emerge for substance use and those that might be quite specific to substance use disorders. And we're utilizing genomic approaches such as the ones that Jordan's group and Phil Lee have spearheaded to address comorbidities between substance use disorders and psychiatric as well as other medical illnesses like insomnia or obesity. So what have we found at the level of individual substance use disorders? Our flagship study was led by Raymond Walters, and it was a GWAS of alcohol dependence. At the time, it seemed like a magnificent sample size. We had nearly 15,000 cases. And what was particularly unique was that we had both a European ancestry and an African ancestry component to this paper. We were very fortunate to have the paper featured on the cover of Nature Neuroscience, including a cover that was designed by one of our co-chairs' daughter. So that's our pretend Manhattan plot. I'm going to assume the glass of beer with a very tall set of drops going out of it is chromosome 4, because that was our key finding. To those of you who follow the literature in terms of the genetics of alcohol use disorders, you might be disappointed to know that what we identified was the functional variant in the alcohol dehydrogenase ADH1B gene, RS1229984, that has been just studied for a very long time in terms of its role in the metabolism of ethanol. But there were some other very interesting insights that emerged from this paper. The first was that by utilizing our African American sample, even though it was smaller and had been studied before, particularly by one of our co-chairs, Joel Galanter, we found that there were two distinct signals within the same gene, ADH1B, one in red that we identified predominantly in our European sample, and the one circled in blue that was identified in our African subset of the data. And interestingly, the LD structure in this region across the ancestral groups was also different. In addition to just the independent loci that we identified, the genetic correlations began to give us some insights that really solidified our rationale for distinguishing between substance use and substance use disorder. So in this panel, you notice that you see the typical genetic correlations with marijuana initiation, smoking, nicotine dependence, smoking cessation. And then you come down and you notice that these were from some of the earlier studies of alcohol consumption, that the genetic correlations between alcohol use disorder and liability to alcohol consumption were modest to high, which made you wonder, why is there so much variability in these correlations? And then are we really studying the same thing? And hopefully I can answer some of that as we move through this talk. The other thing we noticed were the predicted genetic correlations between alcohol use disorders and the other psychiatric disorders and traits that have been studied. Obviously, strong genetic correlations with major depressive disorder, ADHD, depressive symptoms, and a pretty robust genetic correlation with educational attainment. And I'll pause there for one moment to make a comment. The genetic correlation with educational attainment really has proved to be an interesting finding, in that when you look at alcohol use disorder, whether in the Walter study or other studies that have since been published, that genetic correlation is consistently significant and negative, in that liability to lower educational attainment is correlated with liability to alcohol use disorders. In contrast, if you look at some of the GWAS, the very large GWAS of alcohol consumption, you notice that that correlation is either non-significant or significant, but in the opposite direction. And you'll see this one more time as we walk through the slides. So in some ways, the Walter study was a springboard for understanding the role and the place of alcohol use disorders within this spectrum of psychiatric illness. There is now a much larger GWAS of alcohol use disorder and problematic alcohol use. I'm not going to go into that, but just give a shout out to Joel Galanter, who will be presenting on the Million Veteran Program, work by Hang Zhou that is now in press in Nature Neuroscience. So please come back tomorrow to listen to that talk. But let me move on and tell you about our second GWAS that was published earlier this year in Molecular Psychiatry. This one was led by Renato Polimanti, and was a GWAS of opioid dependence. We started out with a very small number of opioid dependent cases. These were the data that truly were available when you were looking at diagnostic opioid dependence. There are other efforts looking at heavy opioid consumption, and another GWAS looking at opioid use disorder out of the MVP that Joel will talk about tomorrow. The PGC opioid dependence GWAS identified two genome-wide significant signals, but what I want to draw your attention to are our control groups. Renato actually used two very interesting control groups. He had a smaller set of opioid exposed controls, and he had a larger set of opioid unexposed controls, individuals who either were unassessed or had not used opioids ever in their lifetime, whereas the exposed controls were individuals who had used opioids. These could be prescription opioids or illicit or recreational use. And we had two genome-wide significant signals, one in our African data subset in a comparison of the unexposed controls to the exposed controls, the unexposed controls to the opioid-dependent cases, and then another one in the European subset. But when we dug deeper, what we found to be really a key component of this analysis was that how we define our controls for substance use disorders actually made a significant difference to the way we were finding our signals and what they meant. So Renato conducted a polygenic risk score analysis looking at the largest GWAS of risk-taking. And what he found was that when you examine polygenic risk scores for risk-taking, not only did they predict opioid dependence, as you see by the little red circles and the blue circles, but they were also significantly associated with opioid exposure, suggesting that when you look at unexposed or unassessed controls, you may inflate your correlations with phenotypes such as risk-taking, those early stages of impulsive drug seeking that actually are confounded when you don't distill your controls down to individuals who may have had the opportunity to use the substance and may have actually experimented with the substance at some point. And this has really influenced our thinking in terms of how we in the future code controls, whether it's for alcohol, which is a very common substance where exposure may not be as significant, or looking at the more addictive substances where exposure and the conditional probability of moving on to dependence may be much higher. We have a third GWAS that's in the works. We're about ready to send this out at the end of this month. This one is led by Emma Johnson. This is a GWAS, which is a collaboration between PGC, IPSYCH, and DECODE of cannabis use disorder. We have 2 genome-wide significant loci. And again, a very interesting set of distinctions between cannabis use and use disorder. The 2 panels you're seeing here look at these in somewhat different ways, but both of them are trying to make the same comment. I'll start with the panel that has the blue circles and the red circles. This is actually a polygenic risk score analysis led by Alex Hatoum. And he examined whether polygenic risk scores for cannabis use from a prior GWAS by the International Cannabis Consortium, when contrasted with polygenic risk scores from the current cannabis use disorder GWAS, were associated with white matter volume in about 4,500 children, 9 to 10 years old, who've never been exposed to marijuana. This includes in utero exposure, passive exposure, as far as we could tell. These are children who are basically drug naive. And the correlations, kind of the associations kind of speak for themselves. Cannabis use disorder has a very small association with reductions in white matter volume in this population. Cannabis use, on the other hand, is not statistically associated, and if anything, veers towards the opposite direction. And if you look at the other panel, which is your traditional genetic correlations using LD score regressions, you'll notice that we see the typical genetic correlations that you'd expect to see with cannabis use disorder. You see it with schizophrenia, with major depression, a very robust genetic correlation with ADHD. You see small effects with anorexia nervosa, and there's more to come there. But you also begin to see the same distinctions that you saw for the other commonly used substance, alcohol, in that if you look at educational attainment, you'll see that the triangle and the circle kind of diverge from each other, and that the genetic correlations are both significant, but when you compare cannabis use liability to cannabis use disorder liability, those correlations genetically with educational attainment tend to go in the opposite direction. And you see somewhat of a same effect for some of the other phenotypes when you walk through the list. For things like SES, they tend to go in the same direction, but for some other things, they tend to deviate and diverge. So we also, as part of our group, are interested in looking at the comorbidity between substance use disorders and other psychiatric and physical health phenotypes. We've engaged with a number of the other PGC groups and hope to also contribute to Jordan's cross disorder analyses. We missed just barely the last run of the cross disorder analysis, but are very much looking forward to being part of the next one. So I'm going to give you some very quick snapshots in the interest of time of what our group has been working on. One of our first collaborations was with the major depression subgroup. And this paper was published in Psychological Medicine, looking at alcohol use disorder and major depression, and evidence emerged for some causal effects of depression on alcohol dependence, but not the other way, although that might change as sample sizes increase. Melissa Manchernoff is spearheading a collaboration between the eating disorders subgroup and the alcohol and substance use disorder subgroup, and she published a paper earlier this year in Addiction Biology that finds interesting genetic correlations between alcohol use disorder, cigarette smoking, and cannabis use with anorexia nervosa. But interestingly, these correlations, these genetic correlations, tend to run differently based on whether you're looking at binge eating or you're looking at anorexia without binge eating. So for instance, the finding was a positive genetic correlation between cannabis use and anorexia, but it was significant only when you looked at AN with binge eating, whereas the genetic correlation with smoking was negative, but only with anorexia nervosa without binge eating. And Melissa is now looking at a cross-disorder analysis with alcohol use disorders. We've also put a lot of effort into looking at alcohol and schizophrenia. This is work being led by Emma Johnson, and she's about ready to circulate these analyses to all her co-authors. Emma used ASSET to combine summary statistics from an alcohol use disorder GWAS that uses the MVP data and the schizophrenia 3 GWAS. And she has several convergent and divergent variants. What's interesting is the signal that ADH1B has, which is strongly associated with protection for alcohol use disorder, is actually also associated with schizophrenia. And finally, just to note that the genetic correlation between alcohol use disorder and schizophrenia is enriched in the brain, but the genetic correlation between drinks per week and schizophrenia is actually fairly small, barely significant, and does not actually show any enrichment. So in terms of our next steps, we're very interested in moving towards polysubstance phenotypes. We're also going to be delving into some symptom level analyses and looking forward to our next iteration of the alcohol use disorder GWAS. And thank you very much. I'll stop there and happy to take questions either here or in our breakout rooms.
Thank you so much, Arpana. That was wonderful.